Hi everyone. In case this is your first time meeting me, I'm Anastasia Chinecherem Ogbozor. Welcome back to our series on the modern tools of biotechnology. In part 1, we discussed PCR. In part 2, we talked about DNA sequencing. And today, in part 3, we're looking at recombinant DNA technology. So, what does this mean? Recombinant DNA technology is simply the process of combining DNA from two different sources to create something new. In other words, scientists cut and join pieces of DNA to give an organism a new trait or function. How does it work? Here's the simple breakdown. 1. Scientists identify the gene they want. Let's say a gene that makes insulin. 2. They cut that gene out using special tools called restriction enzymes. 3. The gene is then inserted into another DNA molecule, often a circular piece of DNA called a plasmid. 4. That plasmid is put into a host cell, usually bacteria. 5. The host cell multiplies, and with it, the new gene is copied and expressed. This is how bacteria can be programmed to produce human insulin for diabetic patients. What are the applications? In medicine, it allows us to produce insulin, vaccines, and even growth hormones. In agriculture, recombinant DNA helps create genetically modified crops that resist pests, tolerate drought, or have higher yields. In research, it's a tool to study genes and their functions. Why does it matter? Because recombinant DNA technology was one of the first breakthroughs that truly showed how we can directly manipulate genetic material. It opened the doors to modern genetic engineering and biotechnology as we know it today. So in simple terms, recombinant DNA is like genetic mix and match, giving us the power to solve problems in health, food, and the environment. Do well to follow me for more simplified science content. And don't forget to like, share, and subscribe, because in part 4, we'll be exploring one of the most exciting tools in biotechnology today, CRISPR-Cas9.